biology using first principles. So these set of methods are called ab initio methods. So ab initio is a Greek word which essentially suggests that you are starting from scratch and you are essentially using some basic ideas about biochemistry and biophysics, or some basic design principles that would be used to figure out some problems in biology using and when these methods are applied using computation, then these are essentially called ab initio methods in bioinformatics. So there are some problems, obviously, which cannot be solved otherwise. So problems which have a composite pattern of information. So like in biology, there are multiple layers of information. There are monomers, there are polymers. These polymers are interacting with each other in different networks. So this is a complex composite pattern of information, and there is no clear underlying rules. There are clear underlying rules, but there are no clear underlying principles in terms of how these composite patterns are working. There are problems where no prior database of exact biological pattern is present. There are problems, obviously, for such composite problems. So these are like complex problems. Most simple algorithms fail to provide an accurate solution. And there are problems where the rules or principles can only be identified using statistical analysis rather than some basic ideas of how things are working. So for example, two such problems which are very important in the context of biology is how do you predict genes in a new organism? So this actually goes along the fourth problem that we had in the lab yesterday. And the next problem is how do you predict the structure of a new protein? This goes more similar to the last problem that we had in the lab yesterday. So in today's class, I'm mostly going to talk about the first problem, which is how to predict genes in a new organism. And in the next class, in next week onwards, I mean that's the last week. I'm going to touch upon how to predict the structure of new protein and talk about some ideas in structural bioinformatics. So when we want to find a gene in an organism, there are two basic approaches. The first approach is called the extrinsic approach, something that you've already been exposed to using, say, homology-based searching. So homology is the idea that a gene is inherited from inherited between organisms. So if if you have a database of genes of one organism, then you can use this database of genes to search for genes in a completely unknown organism. So you did that in the lab. Um, I think problem 2 was based on this question. So these extrinsic methods are essentially using some sequence similarity approaches to find whether a gene exists in an unknown sequence. The other approaches are called intrinsic approaches, which are based on ab initio methods, which essentially work on defining a gene based on certain rules. So they can be divided into two parts. First part for genes that are in prokaryotes and gene finding in eukaryotes, and basically how such methods work. And in this context, I'm going to introduce you to the idea of Markov modeling or Markov models, which are applied to such questions. So before I begin, what would be the difference between a prokaryotic and a eukaryotic gene? Why do you think we need two different methods of approaching this problem?
In prokaryotic gene, there is no like in exons and introns. Those genes are like from a start
Yes.
to end, they are present there. And when eukaryotic comes, we have exons and introns.
Okay,
So,
that's one. What else? If you look at it from the context of the entire genome of a eukaryote versus entire genome of a prokaryote, are there certain
So,
information
so
about?
you have chromosomes in eukaryotes, and but you just have like a singular entity of DNA in prokaryotes.
So does that make a difference? I mean, if you put a
But in prokaryotics also, like there are chromosomes, right? We say
in
that chromosomes.
prokaryotes also there could be like your nuclear chromosome, your plasmid chromosome. So you can have like discontinuous sequence information from prokaryotes as well. So that's not really that much of a difference. What else?
Sir,
Have
in
you heard
UK,
about gene content?
sir, in eukaryotes, a single gene can code for multiple proteins, whereas in prokaryotes, a single gene codes for a single protein.
I think, yeah. So I think Ratnesh already talked about that. The fact that they can do that is because they have introns and exons. So do you know something about the G value? The G value paradox? Were you
Yes,
taught about this?
yes, sir.
Yes, sir.
So can you somebody tell me what that is?
Like number of genes in an organism doesn't define its complexity, something like that. Uh,
Exactly. So
or
now,
something there is C
so
value also, and C value and G value. Yeah.
yeah. So for prokaryotes versus eukaryotes, is there a difference? So do prokaryotes, are they more gene dense versus eukaryotes?
Yes, sir. In prokaryotic
Yes,
genome,
sir.
like almost in E. coli, total the E. coli genome is made up of genes. There are very few parts of this, very few percentage of the genome is non-coding.
Exactly.
On
So
the other
that's
hand, in eukaryotes, 95% of the genome is non-coding. Only 5% is coding.
exactly. So therefore, there would be different principles that we would be applying for both these problems. So we're going to talk about these approaches first, and then I'm going to show you how these approaches work. So the extrinsic approach, it's basically similar to homology modeling. So you take a tool such as for multiple sequence alignment, such as BLAST. If a sequence is similar with another known gene, then it must be the same gene. Um, the issues that you have here is how do you identify which genes align, because you might have two genes which have very similar E values, right? And once you know which genes align, you have to apply repeat BLAST for over 1,000 to 2,000 times, because for every DNA fragment that you would have in your new organism, you would have to put that DNA fragment into the BLAST algorithm, because typically the BLAST algorithm, if you want one gene for every piece of DNA you have, you would have to, you can't put like a large, like you cannot put 10,000 or 100,000 base pairs in the BLAST, because then it will give you multiple locally aligned sequences. So this is going to be a challenge in this. Intrinsic approaches, they basically use rules based on statistical sequence features of a gene. So a gene must follow rules based on a gene's model definition. So the issues that we would have for this is that you would need to know these rules beforehand. So how do you actually define a gene? How does a gene look like? And once you define these rules, you have to figure out a way to efficiently parse or compute which sequence fits this model. So can somebody tell me some of the rules which are required for a gene to be called a gene?
So it should code for a protein.
How do you know if it codes for a protein?
So maybe if it has, like, we can see if it has a start codon and a stop codon.
Okay. What else? So these are the different features that you could use, right? So you would have a promoter. So this is where the RNA polymerase will bind to actually transcribe the gene. Then once you have transcribed the gene, you would want to have a ribosome binding site, also called as the Shine-Delgarno sequence, where the what would bind to the ribosome binding site?
Uh, their ribosome binds
Polymerase.
that
The ribosome
no,
will
sir.
bind to start translation,
Ribosome will bind. Yes,
right?
and that
Uh,
16 RNA.
there would be a transcription start site as well. So this would be, can you tell me where the transcription start site is present on the gene?
It is +1.
The +1 site.
Yes.
And what is 0?
There is no 0 in the gene.
If there's a +1, there's a 0 as well.
No, sir. There is +1 and then there is -1. There is no 0.
I mean, the place where the gene starts, that base is 0 before that, and +1 is just after that. So what about open reading frame? So you said that you could have a start codon and you have a stop codon, but just having those two things is not enough, right? So you might need to have also start codon and stop codon have to be present in frame, right?
Yes.
Yes, sir.
Does that make sense? And then after that, you could also have what's called a transcription end site, a transcription termination site, right? So can you think about what, since we have talked about a lot of algorithms in the
So
course
I had a
already?
small question here.
Yeah.
So are there instances where like a gene can be translated without having like a triplet structure?
Genes can not be translated without a triplet structure, but genes can be translated without a start codon. As in, they can be
So
translated
how is that possible?
from non-standard start codons. This happens specifically for viral genes, which hijack the machinery. Viral genes have something called internal ribosome entry site, called the iris sequences, which allows the ribosome to bind to them and start translation. But the concept of translation, the translation would always be happening through a ribosome, and a ribosome would always require a triplet structure. So it would require 3 bases to which your anticodon will bind, your transfer anticodon present on the transfer RNA, and yeah, and the transfer RNA is going to add to the extending amino acid chain. So triplet structure would be required, but how does that triplet structure and where does that triplet structure begin? That can be different in different organisms. Specifically, viruses, they have very, very different kinds of translation rules. They have to hijack existing machinery, so they hack the process. But the ribosome can only read in triplets. So ribosomes, the machines which make the majority of the proteins, they read in triplets. The case where you have non-template translation, as I discussed with the NPRs, sorry, non-ribosomal protein synthesis, which make antibodies, right, or small antibiotics, not antibodies, small antibiotics. Those don't even require a triplet structure, right? But if you are dependent on the mRNA molecule and the ribosome to make a protein, this would happen. The codes will be read in triplets. Okay.
Fine, sir.
So getting back, I mean, if you have read something else, then please bring it to my attention, and we can look at it together, and I can probably explain it to you. But from the top of my head, it's always triplet structure is always required. So can you think about some of the algorithms that we have discussed in the class so far in the course that could be used to identify, let us say, the promoter sequence? How would you identify the promoter sequence? Any of the previous algorithms that have been taught in
Yes,
class?
these promoters have like a specific data box and that a Pribno box.
TT
So
Raka box.
both
Yes.
backspace
So we can
can be identified using
So-
this just to be the algorithms we learned in last class.
So what kind of algorithm would this be?
Like based on indexes.
Based on indexes. Okay, so what would you be indexing?
the Tata box and the Pribno box.
The Tata box and the Primno box. Okay. And do we know that the Tata box and the Primno box are exactly the same? in all
No,
the
there could be some
Yes,
slight
sir. Yes.
So there
notations.
could be differences as well.
So then what kind of
No, sir.
algorithm, what
Tata box
class of
and
algorithm?
Pribno box are exactly the same. Tata box and TP Gaga box are different, I guess.
Okay, so for something, let us say the Primno box and even the ribosome binding site, which I'm giving you an exact sequence, the Shine-Delgarno sequence. These are, say, conserved. These are the same. So for them, you can make an index, you can make a library, right? Or you can just make a hash table of such sequences and you can just search for them. What about the sequences which might not be conserved exactly across the organisms? What class of algorithms would you use for them?
So you can use the K, T motif search.
So you'll be using motif searches, right?
Yes, sir.
Yeah. So motif searching would be the case for that, right? So now whenever we are trying to figure out these algorithms, having these algorithms, they are only going to be telling you about where this particular motif is going to lie, right? You're going to say, okay, you have a ribosome binding site, you have a transcription start site, you have a promoter, right? But these things don't tell you exactly what information that they are missing. Instead of asking you, I can just tell you that the information that they are missing is they are not telling you exactly how these sequences are appearing, like what is the order of appearance of these sequences, right? Because each time you are applying an algorithm, it's independently going to find whether you have a Shine-Delgarno sequence or whether you have a promoter sequence, whether you have a start codon or whether you have a stop codon, right? But these are all present in a specific order, right? So for example, if you have a DNA molecule and it has an open reading frame, this open reading frame would be present after the promoter, which already, as we've already talked about, it would have a Primno box, it have a Tata box. It will also have transcription factor binding sites. It will have a ribosome binding site. Then between the ribosome binding site and the promoter, there will be a transcription start site, right? And then there will be a start codon after the ribosome binding site. There will be a stop codon up till which the protein translation will happen. And then you will have a transcription termination site, right? So any clues of, can you think about a way in which you could repurpose existing algorithms, let us say motif finding, to solve this problem? Let's go with a greedy
So
method, right?
we
What's the
would have
simplest method?
to,
Yeah,
so
sorry.
probably by storing the value at which you found the promoter sequence or the TSS. In
Okay.
that way, we can probably realize if they're in the order itself.
So you will store the position of each one of these elements and you would check whether they are uh, appearing in the order or not. So basically you will have a list of promoter sequences that you would have found in the DNA. You will have list of transcription start site, list of ribosome binding sites, list of start codons, stop codons, list of transcription termination sites, right? And then you will see which one of them come together. Is that what you want to say? Which one of them are following
Yes,
the
sir.
correct order, right? But
Yes, sir.
but you might know that sometimes if you are reading in the wrong frame, then you might actually get get the wrong codon, right? Or if you might not be in the right, you might have, like, which promoter would be associated with which start codon. That could be a challenge, right? Do you see the problem here?
So, but we can make like three cases here, right? So we start with the first base, then we can start with the second base, then we can start with the third base. And like if you start like from the fourth base onwards, it'll again be like similar to starting with the first base itself.
In the same frame, yes. So, but you would basically need to write lots of if statements, lots of things to check, right? But there are probably faster ways of doing
Yes.
this, right? So what ab initio methods essentially do is they make use of these features such as motifs, sequence composition, sequence biases to predict exactly where in the given sequence you would have a gene and where in that given gene you have the location of these of these features, because each gene could have different relation between where the promoter lies and where the transcription start site lies. So all these arrows, the distances between these arrows could be very different for each gene. So ab initio methods essentially try to predict these things based on coming up with a way to place these things, place these features in a particular order. So how do we, let's start with the simplest case, which is prokaryotic gene finding. So in the case of prokaryotic genes, it's pretty straightforward. You would have a promoter followed by a start and a stop codon, right? So you have a promoter in blue and you have a start codon and a stop codon, right? But you actually, we know that in the case of prokaryotes, because you have this operon structure, so one promoter can actually be followed by multiple start and stop codons, right? So you have, so this is one of the challenges that you might have. The second thing that would be between the start and the stop codon, you would basically be having a potential open reading frame. And for the case of potential open reading frame, as we know that you could have potential 6 different kinds of amino acid sequences that would come about. All right. So when you want to search for a prokaryotic gene, the first thing that you want to do is you want to start search for a start and stop. Then you want to search for promoters, right? And you want to search for genes which are greater than 50 codons long, because most genes are typically having more than 50 amino acids. So this is an assumption. There's always exception to this rule. So when you actually start to do this for prokaryotes, which have usually small genomes, which have high coding density, as we had discussed, that means they have high G value and they have no introns, gene identification is relatively easy. The problems that you usually would be having is the problems of overlapping open reading frames or for short genes and the problem of finding promoters. But if you start to implement the same ideas to eukaryotic genomes, which are much larger, which have much less coding density, you would have problems with intron exon structure. Therefore, the identification rate accuracy is much less. It's about 50% only. And you have many, many more problems. Okay, so a eukaryotic gene finding algorithm, beyond finding a start and stop codon, beyond searching promoters, you would also have to search for intron exon splice sites between each open reading frame, right? And then you would have to come up with the annotation of exactly where the exons and introns are lying. All right, but here we can take advantage of something very interesting in general. Not all codons in an organism are used equally. So have you heard of this term called codon usage or codon bias?
Yes, sir.
Yes.
Yes, sir.
Right? So the idea of codon usage or codon bias is that the same amino acid is coded by multiple codons, right? But the use of those codons, the frequency with which those codons are found in open reading frames is different for different organisms. All right. So now what does this, does this give you some idea? Can you predict whether you are in an open reading frame? If I reverse the problem, that if I give you a gene sequence, if it would be an open reading frame, can you maybe look at its codon bias and predict whether you are in a gene or not, whether you are in an open reading frame or not?
Yes, sir, we can, but that
Yes, sir,
also
we
depends
can.
on a specific organism also, I guess.
That would be dependent on organism to organism. I totally agree. So you, in such algorithms, you need to know beforehand, or you can basically see the codon usage for, let's say, prokaryotes versus eukaryotes. You could say for, if you have prior known information for prokaryotes, a specific class of bacteria, let us say lactobacilli, right? You know the codon usage, you can apply the codon usage principles on a gene fragment, and you can predict whether the part of the gene that you have is actually an open reading frame or not. Right? So here, the first principle that you are using is, and the statistically derived first principle that you're using is that all genes should have an open reading frame, which are going which would be having a codon pattern. And the statistically derived information from this first principle is that not all codons are used equally for each gene. Okay, so for each organism. So there is an unequal usage of codons. This is a universal feature of genomes, and this can very easily help you to distinguish between coding and non-coding regions. All right. For example, if you have 4 codons for the amino acid valine in humans, human exons specifically use the codon GTG 4 times more than they use GTA. So real exons are expected to show what's called codon bias than introns, which would just have a random frequency of uh, triplets. So if you take any version of triplets in your introns, you would essentially find that these are basically not having any bias. So how does this look like? If I plot a gene, right, and these rectangles are exons, okay, you can essentially see that whenever you have an exon, you have a increased codon bias or increased codon usage. Can you come up with a quick algorithm to test whether a particular DNA stretch, what is the codon usage in that DNA stretch? Can you tell me the steps for this?
Sir, also like what is the negative term here of like on x-axis, on y-axis, what is plotted here?
So you're on the y-axis, we are plotting the codon usage. On the x-axis, we have the genes coordinate.
This is the specific graph for a specific codon, right?
This is a specific graph for a specific codon, yes.
So basically we can just take the open reading frame and divide it into triplets between the start and the stop codons, and then we can probably count the frequency of like that specific codon. Or alternatively, if we want to like do it in the entire genome, then we can just divide the genome into triplets and
Okay.
proceed.
So you can basically do this for all the 6 frames, and you can see
Yes,
in
sir.
each of the 6 frames, if you have 64 codons, right? You count for each codon what is the frequency, and maybe you can take um, a median or an average frequency of each of the codons. And if you find that the codons are mostly random, so then this value, the frequency value will be close to 0.25, right? Uh, or if this value, because it's like 4, right? 4 for every amino acid, or if it is not close to 0.2, 0.25, then you essentially have changes in codon usage. All right. So in order to do all these things, besides looking at the codon usage, at sequence features, locating exactly where you have a promoter, where you have all these sequence features, instead of doing these things separately, there is one set of algorithms or one set of tools that you could be using, which is called Markov modeling. Basically, a Markov model describes probability distributions of nucleotides in a DNA sequence, right? And it's dependent on some conditional probabilities of a particular sequence, depending on k previous positions. All right. So what do I mean by that? Whenever you are given a random piece of DNA, right? First, what you want to find in that random piece of DNA is whether you are having a promoter like sequence or not. If you have a promoter like sequence, right, then the next set of DNA sequences should be searched for the remaining features like the transcription start site, ribosome binding site, start codon, like basically this kind of a frame, open reading frame, which is going to have your specific codon bias followed by all these things, right? So once you found the promoter, you expect to find the transcription start site. Once you have found the transcription start site, you expect to find the ribosome binding sequence. After you found the ribosome binding sequence, you are expecting to find start codons, which also may differ, by the way, in different organisms. And once you have the start codons, you might want to see some codon usage biases happening, right? So all these things can be modeled in a single set of like group of algorithms or even tools that are based on Markov modeling. So Markov modeling essentially says that the probability of seeing a specific DNA sequence depends on probability of having seen k previous DNA sequence strings. And I'll show you how this works. Okay, so you have the most basic Markov model, which is called the zero order model, right? Which basically says that my current DNA sequence, the current observation, does not depend on any of the prior observations, right? So it's zero order. So k is zero here. So this represents the random sequence of nucleic acids in a genome, right? So if you were to take all the A's, all the G's, all the T's, and all the C's in the genome, you were to estimate the Markov zero order probabilities, they would just be probabilities for finding an A in the given genome. All right, is this clear?
Yes, sir.
Yes,
Would
sir.
these probabilities always be 0.25?
No, sir. I guess it should not be.
Right? What would it depend on?
Uh, it will depend on like how many codons are for one amino acid and many
No,
things
no,
like.
not just codons. There is something else. There is something even more basic. Is the probability of finding an A in a genome, in every genome, 0.25? If not, then what is that one value that determines?
Sir, 80 is more rich in like
Okay.
80 or GC
But
or like.
is there one value with which the genomes can be assigned to? Like one sort
The
of one
GC
parameter?
content.
Which content?
GC
The
content.
GC content.
GC content, right? So if an organism has high GC content, that essentially means it has high GC. So it will have low AT, right? So depending upon the GC content, you can actually figure out the zero order Markov model probabilities. Now let's go for first order Markov probabilities, right? First order Markov model. So it is determined by the previous state. Only k is equal to 1 here, right? So now first order Markov model actually tells you what is the probability of finding tumors in the genome, right? So this is very good to find GC repeats in your genome. So if you have a first order Markov model, which has been trained on looking for GC repeats in the genome, right? So you take all the locations where you have GCs and you find whether when you are in a region which has GC repeats, what is the probability of finding a C which is preceded by a G. Then you can apply these first order Markov models to find such regions where you have GC repeats. All right. Then you have what's called a second order Markov model, where the current state is dependent on the previous state and the previous state from that, right? So this is going to give you trimer frequencies or three-mer frequencies in the genome. And this is very good for looking at codon patterns, right? This is looking for codon usage, depending on the organism that you're looking at. So mathematically speaking, your zero order Markov model represents probability xi, where xi is each position and x is the value of the nucleotide that you find at that position. First order Markov model is xi given yi minus 1, where xi is the nucleotide found at the ith position and yi minus 1 is the nucleotide found at I minus 1th position. And similarly, you have the zero order Markov model, which is xi given y I minus 1 and z I minus 2, showing you conditional probability of finding xi given the previous two locations have certain nucleic acids. All right. So now let us see how this works. So in order to do gene prediction, you have to train these models. You have to train these models on some existing organisms. So let us say you train these models to estimate some non-random distribution of nucleotide frequencies in the form of probabilities. All right. So if I want to find, if I want to find P of xi, how would I do it? Let us say I want to find P of xi in this sequence. Can you tell me the P of A in this sequence?
Uh, 0.2.
0.2.
It's 0.2. So what was the basic idea? You sum all the A's, right? And you divide by the length of the sequence, right? So basically you estimate, for example, in order to estimate, so similarly, if I ask you to estimate probability of G, what would it be?
0.3.
0.3, right?
Yes, sir. 0.3.
Now, similarly, if I ask you to estimate probability of G given a C, can you tell me? Can you estimate the probability of G given a C?
Yeah, 0.3. That is also.
0.3 again.
Are you sure?
Yes, sir.
So how many tumors you
It's
have?
one, right?
How many tumors you have where the first letter of the tumor is a C?
It should be one, right? Because there are three CG pairs and there are three Cs. So every time there's a C, there's a CG pair.
Exactly. Similarly, if I ask you this, probability of finding a G, given that it was preceded by a and a T first?
0.
Everybody agrees this is 0?
Yes, sir.
Yes, sir.
So when I say we train these models, you give some known sequences. So when I say you'd be training a hidden Markov model or a Markov model, you would be training the models from known gene sequences, right? And you could be training the kn order Markov model based on some prior data that you would be giving to these algorithms. All right. So in simple prokaryotic gene prediction tools, they try to capture hexamer frequencies because hexamer frequencies give you both information of your Scheindel-Carnot sequences. It gives you information about your codon usage biases. It gives you information of about start codons and stop codons as well, right? And so therefore, these are very accurate. So if I were to capture hexamer frequencies, what order Markov model would I be looking at?
5.
What would be the order of the model for hexamer frequencies?
So 6. No, sir, it should be 5, right?
5, 6. How many are for 5? How many are for 6?
So
5.
I say 5.
5, right?
Sir, like, what is hexamer here? If you can describe.
Hexamer means a 6mer. Hex is 6.
Yes, sir.
So kmer, right?
Yes.
So a 2mer is called a dimer. A 3mer is called a trimer. It's just like that. A hexamer means 6. So if you want to look at the frequency of hexamers, you would actually be you will be training a Markov model with, you'll be training a 5-order Markov model, 5th-order Markov model. If you remember,
Yes,
0th
sir.
order is for just
Yes.
this, 1st order is for dimers, 2nd order is for trimers. So basically 5th order will be for hexamers. Okay, so think about how you will be coding a tool where you will be predicting, where you will be estimating the hexamer frequencies in a given sequence. Please think about this code. Okay, so again, how would you be going about it? For example, for finding for training a 0-order Markov model in this particular sequence, you would be extracting all the 1mers, right? And you'll be figuring out these probabilities. For a 2-order Markov model, for a 1st order, you will be extracting all the 2mers, and then you will be estimating the specific 2mer frequencies given in this format. Similarly for all the 3mers. Is this clear? All these things will be
Yes, sir.
spread across your entire
Yes, sir.
sequence,
sir.
and each of these things will be captured and you will be training. So now how do these things actually look like? So these hexamer models are very good for short genes, right? But for typically longer genes, hexamer modeling is not that good. So typically for much longer genes than 100 amino acids, you have what's called interpolated Markov models. So interpolated Markov models, they actually look at, they go for not only a single Markov model, like a single five-mer Markov model, but they have multiple Markov models. They have 1 to 8 order Markov models. So an 8 order Markov model would be looking at what k-mer size?
7 or 8. 9, sir. 9.
Right? Sure, 9.
Yes, sir, 9.
All right, good. So now, how do these models work? These models would essentially have a separate model for, let's say, a start codon, right? So it will be looking for a start codon. And let us say these are the start codon frequencies. So a start codon is typically having 80% of the time it has a A in the first position, 97% of the time it has a T or a U in the second position, and 97% of the times it has a G in the third position of the k-mer, right? But as you can see here, there are also start codons which have a G at 10% of the times, right? And it has a T at the beginning 8% of the times, right? So now when it's this, you apply this Markov, you give this, you have a Markov model like this. So it has two kinds of coding states. There is a typical coding state, which is represented by a specific codon bias. And there's another atypical coding state, which is again corresponds to another codon bias. And you say that there are 10% of the genes would be from my atypical state, and 90% of the genes will be from the other typical state. So these are representing slightly longer codons, longer genes. And then again, you have another model for your stop codon. Now, what happens when you give it a gene? When you give it a a sequence, you give it whenever you give it a sequence, it starts to calculate the probabilities. Let us say your gene starts from ATG, right? So it will extract the first three-mer. It will say, okay, the first three-mer, the A is at 0.8. My second one is at 0.97 probability. The other one, what I've observed is 0.97. So the probability of seeing of being in the start codon state is 0.8 multiplied by 0.97 multiplied by 0.97, which is approximately 80%. So now with 80% certainty, when you say that I am in the start codon state, then what you are going to do is you're going to transition to the next state. You say that if I am with X% certainty, I am in a specific state, then I will transition to the next state. So then it would be looking for probabilities in your coding state. All right. And then when it will be when it's looking in the coding state, when at that point of time, when it starts to extract the codons, it says, okay, whatever state that you are having in your coding state, it will compare the coding state and the next state. When it will seems like that you are in the stop codon state, right? Then it will transition to stop codon state. So how does this actually look like? This actually looks more like a more complex Gedi route that we were talking about in a dynamic programming algorithm, right? So in a dynamic programming algorithm, you could only travel, say, east or south. But in this case, you can travel from each of these three states to any of these three states in a specific pattern. All right. So a HMM diagram essentially looks something like this. So you have three states. From A, you can remain in A, right? Let us say A is a start codon state. From B, you can remain in B. You are still in coding state. And from C, you can remain in C that you actually saw multiple stop codons. Typically, this would mean that once you see a start codon, you will not likely to see a next start codon. So these A to A transition and C to C transition, the probability is going to be very low. But the probability of B to B transition is going to be very high, because once you are in the coding state, you will remain in the coding state until you see a stop codon, right? So the probability is much higher for this. So now the transitions from these different states can be represented by one of these complex graphs. All right. And once you are giving your input sequence, what you do is you traverse this graph, you calculate the probabilities, the best probability in this graph, which represents, which could be represented by your input sequence, and that is what you report. All right. So essentially in this case, I'm showing you that it starts from state B, then it goes to state A, it remains in state A, then it goes to state C, state C, state B. Probably this path is the best path that is represented uh, by the input sequence. All right. So now if you want to implement a hidden Markov model, it's just a more complicated way, basically where all routes are possible. You essentially give the score, transition score, as the max of all the possible states, given the score of the previous state, multiplied by the edge weight of transition from one edge to the other edge. Okay, this is just for your information. I am not going to ask you to code this, but I just want to show you that the fact that you have understood how dynamic programming works, exactly the same algorithms can be implemented to solve the problems of hidden Markov modeling as well. Okay, so there are different tools which are used to implement these Markov models for prokaryotic gene prediction. So GeneMark is one tool which uses a fifth order Markov model. Glimmer is a tool which uses this interpolated Markov model, that is multiple Markov models. Then you have FGene SB, which is again uses a fifth order Markov model, right? Other above tools would give improper prediction of opening frames, as they are not trained to locate ribosome binding sites. So you should always first locate the ribosome binding sites by some kind of a motif searching tool such as RBS Finder. And then once you have found the ribosome binding sites, then within the sequence that follows the ribosome binding site could be fed into these tools. The gene prediction tools for Markov for eukaryotes is slightly more complicated because you have to figure out these intron-exon boundaries. This is how an intron-exon boundary looks like. You would typical when I say typical, these are often called canonical in terms of in terms of sequence language. So canonical means the most typical or the best, most often found sequence for that particular feature. So for example, the canonical start codon is AUG. So typically canonical um, splice site. So the 5' exon donor site is AG followed by GT, right? Followed by, it could be a A or a G followed by a or GT. Then you have an intron sequence, and then you have the 3' donor site, which is going to have a stretch of pyrimidines, 10 pyrimidines, followed by an X, followed by a CAG. This CAG could also be replaced by an NAG, right? Which could be N means any letter. And then this is followed by a G followed by a GT. All right, so you can see that this definition becomes slightly more complicated. This is captured by tools such as GeneScan, which, as you can see, are a more complicated version of interpolated Markov models. Actually, they go into what's called hidden Markov models, where you have these kinds of input transition states. I would not go into the detail of exactly how they work, but the idea basically is that you have a initial transition state, from which you transition into, let's say, your coding sequence state, and these different states have uh, certain probabilities of staying within that state based on this Markov model training. So basically, the donor splice sites have a specific Markov model behind them. Similarly, you will have another Markov model which would be based for a prediction of your codon biases, so on and so forth. All right, and this is what output looks like from a GeneScan. This is what you'll be doing in your lab next in the next class, in the next lab. It will be a mixture of GeneScan and yeah, it will be a mixture of GeneScan and yeah, next lab would be GeneScan, and the lab after that would be AlphaFold, right? So typically the GeneScan is going to tell you at such and such location in the given sequence. So at position 43, ending at position 88, you had initiation sequence, And the probability for finding this sequence, given the transition probabilities that were calculated from the hidden Markov model, let's say was 0.69, right? Similarly, then you have the next feature that it was annotated was an INTR. So an INTR represents an internal exon. You say that the internal exon was found from this location to this location. The length of it was this much. And with this much probability, we can say that this was an exon, internal exon. Then you have the next internal exon, then you have what's called the terminal exon. So INIT represents the initiation exon, term represents the termination exon, so the last exon. So what are going to be the features for internal exon and what are going to be features for terminal exon? Can somebody tell me? Can somebody tell me what is going to be the one distinguishing feature for an internal, for the initiation exon? And what is
So
going to
it
be
will
one?
be it will have ATG of a start
Yes.
codon.
And
That
what
means
about a terminal exon?
The
It's going to
start
have a.
and it would have the stop codon.
terminal.
And
Yes,
all
sir.
the internal exons are not going to have either stop codons or start codons, right?
Yes, sir.
And then you have what's called like a
Yes, sir.
poly A tail, right? So it says that they found a poly A tail at this location and they would have a specific probability. So typically this software like GeneScan, they would output certain probabilities. So if you have a probability of greater than 0.99, that means you are 100% sure that the predicted feature, you say, for example, in this case, you have this 0.97, right? That means that it's very high probability that you actually have an exon here. And if the probability is less than 0.5, then it basically means that it's not very sure whether the feature that is being predicted is the correct feature or not. So GeneScan also uses a fifth order hidden Markov model along with like interpolated IMM model, which basically detects your splice sites and it is trained on vertebrates. So all the organisms which have a backbone, it's trained on Arabidopsis, which is Arabidopsis and maize, which are two model plants which are used. Yeah, and its accuracy for predicting coding nucleotide is more than 90%. Its accuracy for predicting splice sites is about 70 to 75%, and accuracy for predicting complete gene is only about 50%. So as you can see, predicting genes in eukaryotes is much tougher. So therefore, there are new tools which are coming up, which are based on machine learning, which either use things like support vector machines or deep learning. Again, the principles are the same. You give an input data set of genes, which of known genes, and it learns features from these known genes, these tools. And after learning features from the known genes, these tools then apply these predictions on a new input sequence. All right, just like we did for hidden Markov model. Hidden Markov model has these kinds of probabilities calculations, right, of appearance of certain k-mers. But in the case of SVM and deep learning, you have some other. So in the case of deep learning, there is no k-mers, but there is like a neural network of a specific size that you say, which are also going to be something similar but it's like slightly a more complicated hidden Markov model. And then you have a support vector machine where you have slightly more user-defined features that you can go and read about and ask me questions if you have any. So I think I'll end the class here and take questions if you have any for this.